Hi guys, my name is Anna Urena. This is my presentation on adhesion junctions. So to give you an overview and general information, in animals, one of the most common kinds of cell-to-cell junctions are adhesive junctions. The main function is cell-cell adhesion. There are two types of adhesion junctions, which includes adherence junction and desmosomes. Adherent junctions is the intermediate membrane features includes continuous zones of attachment, and desmosome is the intermediate features includes localized points of attachments. Adhesive junctions link adjoining cells. They do so by linking cells together into tissues, thereby enabling the cells to function as a unit. They anchor the cytoskeleton to the cell surface, in which this cytoskeletal network helps maintain tissue integrity to withstand mechanical stress. Adhesive junctions rely on specialized adhesion proteins. Many are transmembrane proteins, meaning the extracellular portion of these proteins can interact with the extracellular portion of similar proteins on the surface of neighboring cells. Figure 1 is an image of epithelial desmosome. The structure and proteins involved, as previously mentioned, the two main kinds of cell-cell adhesive junctions are adherence and desmosome. The associated structure for adherence junctions include actin microfilaments, and the associated structure for desmosomes includes intermediate filaments known as tonofilaments. They're button-like points of strong adhesion between adjacent cells in a tissue. Despite the structural and functional differences, they both rely on intercellular attachment proteins and cadherins. Adherence junctions, or cadherins, the beta-catenin is a protein that binds to the cytosolic tail of the cadherin. Roles include cell adhesion, functions in WNT pathway, which is cell signaling pathway important in cancer. The alpha-catenin is the second protein in which beta-catenin binds to. It can recruit actin to the junction. And desmosomes rely on cadherins, which are called desmocollins and desmogloins. The beta-catenin in this protein is a family protein plakoglobin, binds to desmocollin. The location of physiological function and role. Adherence junction are found in many epithelial cells, or E-cadherin, which are types such as the lining of the intestine, kidney cells, and epidermal cells. They contain plaque, which attaches to the membrane proteins and microfilaments of the cytoskeleton, and they rely on cadherins and their associated proteins for adhesion between cells. Desmosomes are found in many tissues, but are especially abundant in cells that experience mechanical stress, such as the skin, heart muscle, and uterus. And as previously mentioned, their button-like structure binds cells together and functions as anchor for fibers on the cytoskeleton. Figure 4 is an image on the major types of cell attachments in epithelial cells. Diseases that are associated with the malfunction is that there are several, actually, which includes loss of desmosomal components. This could be a very devastating condition. An example of this is mice lacking plakoglobin that die with heart failure and skin defects. Mutations in desmocollins expressed in the heart can lead to damage to the heart muscle and in an adult human patient. Human patients who developed autoimmune reactions against components of their desmosomes or who have mutations in desmosomal proteins often develop blistering diseases of the skin. Cancer cells often stop expressing cadherins, such as E-cadherin, on their surfaces, resulting in a reduction of cadherin expression and then spreading to other parts of the body. The process is known as metastasis, which is a term referring to cancer spreading in multiple parts of the body. This was an article that I found that has to do with the loss of E-cadherin promoting metastasis via multiple downstream transcription pathways. Loss of E-cadherin is thought to enable metastasis by disrupting intercellular contacts. Gene expression analysis shows that E-cadherin loss results in the induction of multiple transcription factors, at least one of which twist is necessary for E-cadherin loss-induced metastasis. In human tumors, loss of or reduction of E-cadherin expression can be caused by somatic mutations, chromosomal deletions, proteolytic cleavage, and silencing of CDH1 promoter. With this research, it was found that multiple transcriptional pathways are activated on E-cadherin loss, at least 2 of which contribute to the later steps of metastatic dissemination. Figure 5 is data that was obtained from this article supporting the fact that loss of E-cadherin is sufficient for metastasis, whereas cellular disintegration is not. And questions in regards to my presentation was that I want to know what's the most interesting thing you learned about adhesion junctions and what would interest you for potential research with loss of E-cadherin.